Well, hello everybody and welcome to Coffee Chats with Experts. I am your host, Laura Moriarty. And welcome to today's Coffee Chat, which is going to be a discussion on the topic of real-time PCR in infectious disease. And as usual, I'm very pleased to introduce our esteemed panelists for this morning. So first up, we have Brandon. Hi, Brandon.
Good morning.
Good morning. So Brandon is based there in the Midwest, and he has a background in molecular biology, which is perfect for this coffee chat, and 15 years experience with real-time PCR. And he's been at Bio-Rad for over 6 years and counting, and he's been supporting researchers all across the United States in both the academic laboratories and in the biopharma side of things. So welcome again, Brandon.
Thank you.
And next up, we have Adam. Hey, Adam.
Morning, Laura.
Morning. So Adam is joining us from, I believe, Texas this morning. And he has a background also in molecular biology with a little spin in oncology as well, and 12 years experience with real-time PCR. And interesting fact, during his graduate work, he claims to have assembled as many 96 well plates and started seeing them in his sleep. So I think that sounds pretty reasonable to me, having done a lot of 96 well plate assembly myself. So hopefully this doesn't bring back too many bad nightmares for you today. So he's been at Bio-Rad for 5 years now, and he serves as the application lead for both qPCR and gene expression. And as a field application scientist, he's been also working with researchers in the US, in academia and biopharma too. So great to have you with us, Adam.
Great to be here.
And then finally, we have Jan. Morning, Jan.
Hey, Laura.
Hey. And so Jan has a background not just in molecular biology, but in business. So Jan's been at Bio-Rad for 11 years, and he's actually based in California, same as myself. And he has, as part of his R&D career, been focused for several years on pathogen detection. So actually a great person to have on the call since we're talking about infectious disease. And he's been working both on the biopharma side of things as well as on the clinical side of things. So, and he's also launched quite a few products while working at Bio-Rad. So I think this morning we're quite lucky to have this great, well-rounded panel of people. And before I kick us off with some questions, I just wanted to do a couple of housekeeping things, and that is to please submit your questions. We love to get your questions, so put those in the Q&A. And we will have our panelists answer them. But not to worry, if you're listening on demand, you will also get your questions addressed, and we will respond back to you through the email address that you have given. Okay, so is everybody ready?
Yeah.
Uh-oh. Yes? Okay, thumbs up. I like the thumbs up. I like the thumbs up. That's good. All right, so as always, I like to kick us off with a couple of questions to warm everybody up. So here's the first one. And honestly, if you can't answer this, then you shouldn't be on the call. So for the panelists, what is qPCR and why is it useful in the detection and research of infectious disease? Who would like to take this one? Okay, go for it.
So qPCR is a technique that allows us to monitor the same classic PCR reaction we've done for years, but in real time by attaching a second piece of information, which is a fluorophore that increases in brightness as we accumulate additional amounts of DNA amplification. So it is really allowing us to take the number of cycles that are necessary to amplify a target and understand the starting amount of material. And this is very useful in studying diseases and infectious diseases because it's highly specific and can identify things with remarkably low amounts of starting material. So something like, say, the SARS-CoV-2 genome can be found with a great deal of understanding that it is, in fact, that genome and that alone and not another target, and also at exceedingly low levels that might be found during parts of the infection.
Beautiful. Couldn't have said it better myself. All right. So that's one person warmed up. Let's see if we can get someone else going here this morning. All right. Oh, this one I love because this one could just take up the whole coffee chat. But let's maybe keep it to the few key points. And that is what makes for a good qPCR assay. Go for it. All right.
So yeah, I'm trying not to take up the whole chat, but a couple of key things that you want to look for when designing and validating a qPCR assay are efficiency and specificity, right? So efficiency, when we run qPCR, in theory, the PCR amplicon should be doubling with each cycle as we take it through the cycling protocol. So an assay that's 100% efficient would be one that's perfectly doubling. Now, we're all biologists, so we know that nothing ever works out perfectly. What we kind of shoot for is something give or take about 10%, right? So between about 90% and 110%. And that can be determined by validating the assay, running a standard curve, making a serial 10-fold dilution, and using the slope of that best fit line to determine what the PCR efficiency is. So 100%, something pretty close to that. The other thing we want to look for is specificity. We want to make sure that our PCR primers are amplifying only one specific target, the one we're looking for, right? Anyone who's ever run a PCR gel enough times knows that sometimes you'd run that lane on the gel and you might see these extra bands showing up. And if you're running a gel, that's maybe not such a big deal because you can just ignore them, but SYBR Green is going to bind to those extra products if you have them. If you have a primer dimer, then that could be confounding your data as well. So you want to be running a melt curve if you're running SYBR Green to ensure that your primers are amplifying only one specific product. Maybe a more old-fashioned method, some people run post-PCR gels on after their PCR to make sure that they only have one band showing up. And then if you want to go above and beyond, some have even next-generation sequence their products to ensure that they're getting what they're looking for. So those are the two main things, are efficiency and specificity. And
I love that you threw in primer, dimer. It makes me think of a Dr. Seuss book. With all the rhyming and everything. So I think I might introduce that one to my children today. All right, here we go. So from the audience, here's a good one. Ooh, what is the smallest amount that qPCR can detect? Jan?
I'd like to, yes. So in theory, qPCR can really detect single molecules. That has been done in certain publications, and the reality is the science backs it. We could detect a single nucleic acid molecule in a reaction. Most reactions, though, don't determine the limit of detection of an assay that way, and they basically establish that limit of detection by the lowest amount that can be detected with 95% typically certainty. And that also is based on the amount of false positives that could arise from their no template reactions. So, long story short, in theory, single molecule, in reality, closer to, I would say, 10 to 5 molecules per reaction. That has been the standard that has been claimed, at least by most pathogen detection assays. And it is very well accepted that if you can detect 10 molecules per reaction, you are at the lowest limit of detection by this method.
Beautiful. All right. Thanks, Sean. Let's take us on to another audience question. Oh, here we go. So what are the conditions that I need to consider while designing a PCR primer? Ooh, who would like this one? Go for it.
The conditions in the case of the reaction itself are making sure that you have like you said, primer dimers. You're trying to make sure that your primer does not actually just bind to itself, that it has a TM that's roughly comparable to what you're going to be running the reaction at. So most reactions these days are running in the 55 to 60 Celsius range. And what we're hoping for there is that it be just warm enough for the primers to bind, but not so much that if you say had a high TM for it, they would bind to sites that were non-cognate, the off-target effect of binding to something non-discriminately. So above all else, the condition that matters is the TM at which the primers bind and the amplification occurs. I would say the GC content and the hairpinning of that are the other components.
Anything else, Jan?
Yeah, I'd like to add one more thing to keep in mind when designing PCR primers is the naming, the nomenclature. I mean, as you start in PCR, you might be tempted to name your primers 1F and 1R, thinking that you're going to be able to keep track of all of that once they're in your freezer, but that is delusional. Start by having a nomenclature first that can be, you know, small enough, but that can be informative enough, like BACT 1F or BACT, you know, 256R, whatever you decide to choose, whether the position in the gene or anything. Just don't make the same mistakes that I made when I started doing qPCR, and I left to my successors a completely ununderstandable system and classification. So there you go. That's my tidbit on that.
I feel like we should also then be having a coffee chat about experimental design and, you know, freezer best practices for using -80 freezers. Because I think we've all suffered from that in the past. Sorry, did anyone have anything else to add on primer design?
Yeah, Laura, I'll tack on one thing. So yeah, nomenclature is definitely important. So thank you, Jan. One other thing that I would add to that is that, you know, the questions about design, there are a lot of good design algorithms out there, but just keep in mind that you never really know how an assay is going to perform until you do it in your lab under your conditions with the mixes and the conditions and the machine that you're running. And so it's important to validate and optimize primers after you get them, right? You can run a thermal gradient to optimize that TM, or you can also buy primers and assays that have already been validated. Bio-Rad has those with prime PCR assays.
Yeah.
Brilliant.
The other, I'll chime in on that, but the other thing to keep in mind when designing PCR primers, like Adam said, you could buy them pre-designed, and what you get there is the certainty that all the assays in that collection have been designed with the same conditions. You need to keep in mind that you may want to multiplex your qPCR reactions at a later time. So if you're starting by the detection of one target, you might think it doesn't matter what conditions, I could have, you know, a melting temperature of 65 degrees and be perfectly fine. But if all your other assays are optimized for a melting temperature of 60 degrees, you will find it troublesome to multiplex those assays. So it is good to develop a system and to keep in mind that at some point you will have to, or you will be tempted to multiplex. And having similar conditions, similar temperature really help with that as well.
Awesome. All right, let's move us along. So here is, oh, this is a long one. So this person has 4 samples, all are reverse transcribed with about 1 microgram of RNA into cDNA. They're taking 1 nanogram of each cDNA, running a housekeeping gene, using FAM probes. Why are the CT values for all samples not exactly the same, since the starting input was the same? I'll give you a second.
I'll take this.
This is a good one. Go for it.
Yeah, so there's always the underlying assumption that we can truly measure the amount of DNA within a sample using something like either a fluorimeter or a spectrometer. That number is reliant on either how much light is bounced off of a target that's fluorescing or how much passes through a sample that's occluding it because there's DNA there. That's an incredibly rough estimate, and it's assuming a fixed amount of every single equal amount of those genomes or different DNA that you might be working with. You can get within a very close range, but expecting to be able to extrapolate a spectrophotometric concentration directly to copies of DNA is unfortunately not going to carry over. So it can be off by something on the order of, say, plus and minus 5-fold. If you end up within 1 or 2 cycles with this kind of data, that's a testament to you doing everything right.
I'd like to chime in on that one too, and add the fact that pipetting really is never as accurate as it says. I mean, you might be measuring on your nanodrop or whatever, what you estimate to be 1 microliter of your solution. The reality is it could be 0.75 or 1.2 microliters. And the same thing applies then when you go on to performing your qPCR, and you think that you're diluting everything at the same amount, but there is some variability. And that is really why we use housekeeping genes. And this is why you're performing your experiment well. Housekeeping genes help us normalize that and really find out the differences in gene expression levels. The absolute CTs, like Brandon said, if you get within 1 CT of the same of the samples at the same concentration, you've been doing things extremely accurately. But the reality is, when you normalize things over your reference genes, and I do specify genes because it's always better to use more than one, then you're even more certain of getting reproducible results.
Great advice. All right, let's move us on. Oh, I think this is a good one as well. So, and I think we have the right people on the panel to address this. So how does qPCR compare to ddPCR, which I'm in R from the Bio-Rad point of view, it would be droplet digital PCR in terms of utility in pathogen detection applications? My goodness, I can't speak this morning. Who would like to kick us off? Go for it.
I can take that one, Laura. So qPCR and droplet digital PCR are both great technologies, and each of them kind of have their own set of advantages. So droplet digital PCR is obviously a technology we're really into at Bio-Rad. It has excellent sensitivity and precision because it's partitioning the sample and counting the number of DNA molecules that you have. When you compare qPCR, one big difference is going to be the throughput and time to answer, right? qPCR, you can set up and run a plate. It'll take you an hour and a half. If you're looking for a positive, negative signal, then that is going to, detect whether or not you have something. The workflow for droplet digital PCR is going to take more along the lines of 5 to 6 hours. And so if you're looking for absolute quantification, great precision, then that's going to be a tool that will help you with that, but it is going to take more time and have a lower throughput.
Yeah, and to add on to what Adam said also, digital PCR offers a great advantage in that the quantification is much less sensitive to the presence of any PCR inhibitors. And this is particularly useful when you're testing raw samples, for environmental studies, for instance, where you start with very crude material. This can definitely help level up your quantification. Now, in most molecular assays, I would say that you start from a relatively clean sample. If you've purified it by columns or some other method, typically your samples don't have those sort of problems, so you should be fine. One advantage I would say for qPCR in this case is, on top of the throughput, you have a little more flexibility in terms of the input as well. You are not limited by the volume of the reaction, whereas most digital PCR reactions really only allow you to put in 10 or 5 microliters of sample to start with. You can do a qPCR reaction with up to 150 microliters if you wanted. If you were using a deep well instrument with a 4x mix, you can really put in a lot of sample and therefore increase your limit or decrease your limit of detection, I would say.
So definitely pluses and minuses to both technologies. It seems like somebody would really need to be thoughtful about all of the different parameters that you've spoken about just now and identify which one is going to work best. So I think that that's great. Okay, so let's move us along. Okay, so if you run a RT-PCR for 50 cycles, for example, would you accept as positive a sample with a CT value of 49? How would you suggest to set thresholds and baselines? Who would like to take a stab at this one?
I can do that.
Go ahead.
I'll just say this. Any PCR, given enough cycles, will amplify. Every PCR is always amplifying some other target at an exceedingly low efficiency, and it appears not to occur within the 40 cycles we typically run a PCR because it hasn't had enough time to become a fully manifest species within that. But anything after about cycle 38 for an efficient PCR is exceedingly spurious. We don't want to put a lot of faith in that, because if we were to back calculate from there to the starting concentration, after cycle 38, it's less than a single integer copy of DNA. And that can't happen. The minimum piece of DNA you could have is 1. So anything after cycle 38, you would have to have proven aggressively using a standard curve and LOD, show that perhaps it's a low efficiency assay, but you can find things into the 40s. But by the time something is in the 40 cycle range, you are going to have to put a lot more effort into proving why that was able to work, rather than being able to simply run with it just like any other lower CQ or CT. As far as the thresholds and the baselines, they don't really feel any different after cycle 49. You would simply be taking the samples that you had seen before cycle 30 or so and extrapolating those out, and those would just be at the inflection point of the amplification plot. So as long as you have a sigmoidal amplification plot and you see an inflection point between the exponential and linear phases, it wouldn't matter how late it came up, but it would be very suspect without a lot of controls.
Fantastic.
Oh.
Go ahead.
I think they want to chime in.
Yeah, I think so.
Go ahead, Adam.
Oh, yeah. So that kind of highlighted another point that when I was talking about running a standard curve earlier, another thing that a standard curve serves to do, apart from help you determine the efficiency of the reaction, is to establish the dynamic range of the reaction, right? If I just keep diluting my standards over and over again, that standard curve line isn't going to go on forever, right? At some point, you're going to lose linearity if you dilute your sample enough. And so it can help you establish the dynamic range, where you can say, okay, anything from a CQ value of maybe 18 to 31, I can count on. But if I get a CQ value of 35, 40, right, I can't assume that that's within the dynamic range. And that's another reason to look for lower limit of detection. And then with regard to thresholding, the good news is that most softwares, CFX Maestro being one of them, are going to set the threshold for you automatically using an algorithm in line with what the best practices are, right? To look for sort of the end of that exponential phase of the curve, it uses the maximum of the second derivative and sets the threshold there. And that's where you see that linearity between the log of the starting quantity and the CQ value.
Jan?
Yeah, and I'll chime in on that. I mean, it's I think RT-PCR definitely is a method that allows for a lot of elasticity, and I think your experimental design should dictate the number of cycles that ultimately you rely on. If you're working with ancient DNA, frozen in the tundra that is hyper degraded, and you're just looking for that one copy of elusive virus, maybe you can have conditions in which a cycle of 49 will give you the eureka, I got it, COVID-19 was present millions of years ago, or something like that. But it's for more regular applications, I would stick with Brandon and Adam's recommendations. One little caveat, though, you might probably have seen within the, I'm sorry for bringing it back to it, but it's very present in everybody's life right now, the SARS-CoV-2 detection. Several kids have, several clinical UA kits have thresholds of detection past 40. And it is linked to the fact that you are favoring, in some cases, sensitivity over specificity. And you really want to make sure that you detect any positive in your sample pool, even if sometimes it comes at the expense of some false positives as well. So that is a long explanation that you probably didn't wish you had, but that's what you get for asking the question.
I love it. All right, so here's a nice long question to combat that long answer. Are you ready? Get those reading glasses on. All right, so this person's seen labs that create standard curves with their samples. For 4 samples, they take 5 microliters of each, mix it, and run a housekeeping gene on it with several dilutions to create a standard curve. Then they read samples off of this curve. When they have more samples from the same study, they create another standard curve from the new samples. I don't think this is correct. I would like your thoughts on this too. Yeah, this is a long question. Adam?
Yeah. So I'm not sure I caught exactly how things are being done. I would say that if you're wanting to calibrate your samples to a standard curve, you would want to be running that standard curve on each plate each time you run qPCR, right? Because that's going to account for any inter-run variability, right? Where you may run into problems is, if I have a standard curve over on plate A, I ran it and some samples, and then I took some other samples on plate B and was getting a CQ value for those, but not running a standard curve, trying to calibrate them back to the standard curve on plate A, then I might be running into some problems because, that plate B may have come up a half cycle later, and there may have been some inter-run variability. So that would be my 2 cents, is just if you are needing to run standard curves and calculate your quantities in an absolute manner, then you want to make sure that they're on each plate that you're running the samples for.
Yeah, and I'd like to answer that question. So Lisa, and I don't know if the person who asked the question is Lisa, but I figure I'd speak to Lisa instead. Lisa, if you are seeing that, you can ask your coworker the question of whether they are only interpreting the data within a plate. And if that is the case, then yeah, it's fine to do that because you're only comparing relative expressions from the same plate. If you are having interplate results and data analysis, then this approach doesn't make sense, and you have my blessing to publicly shame your coworker for doing that, like harshly. Okay.
That's a good one. So now I feel like I need to give names to all the people who are putting these questions in. We need a little jar with names in it, or a random name generator, I think, for the next coffee chat. So I feel a trend happening now. So this is awesome. Okay. All right, here we go. Here's another one. And we really don't advocate public shaming, right, at Bio-Rad, but for the purposes of scientific rigor, right, we want people to do the best science they can.
Public shaming is terrific.
Oh, God. All right, here we go. Here's another one. Oh, this is a good one. What are some of the most common applications for qPCR pathogen detect? Oh, my gosh. I'm going to start that again. What are some of the most common applications for qPCR for pathogen testing in food safety, water quality, and environmental areas? Well, I mean, I can give as an example of that. I had a summer job where I was taking oysters, and these oysters were being served at a fancy restaurant, and I had to make a milkshake out of the oysters and grow it up at 37 degrees in some media and do some qPCR testing on that. And we were looking for E. coli in that particular example. So that's one I can throw into the mix. That was a pretty fun job. All right, I'll let the panelists share some of their insights, though.
Yeah, I mean, I think, the 3 categories that you named are probably some of the top ones for qPCR applications. I mean, whether you're testing for legionellosis in air filters, whether you're testing, of course, for foodborne pathogens like Laura did with oysters, for water quality, again, norovirus is a very prevalent target. One funny, not funny at all, but one more recent application of qPCR is really the testing of COVID-19 levels in sewage water. And basically, this is sort of a ballpark estimation for in certain countries, in certain cities, to see whether the levels are increasing or decreasing with compared to previous periods. So that is, you know, a little more current. But yes, there's definitely all sorts of applications. You need to remember also that qPCR is very heavily used for clinical applications. There are a variety of systems that employ qPCR to determine the pathogen in certain infections, in particular when trying to diagnose sepsis and trying to choose which antibiotics to apply to the patient. The rapidity of qPCR and its sensitivity really are what allow to give a fast answer for those type of questions.
Adam, Brandon, anything to add?
I think that was pretty comprehensive.
Yeah, I would just kind of tack on, speaking very generally, that that, you know, if you're looking at trying to detect a particular pathogen, you know, you want to consider what kind of sample you're looking at, right? Is it an environmental sample? Is it, you know, if you're trying to extract something from soil, that would be different from extracting it to blood, right? So you want to make sure that the extraction kit you have is appropriate for that. You want to consider what kind of nucleic acid it is, right? If it's an RNA virus, then you're going to need a one-step kit or a cDNA synthesis RT kit. If it's a DNA virus, you can use a standard kit. And then you just need an assay that you've designed or that's been validated for that particular target.
Beautiful. Yeah, it makes me think maybe we should have a coffee chat with some of our folks on the food science side of things, because they would be able to reel off probably another 20 or 30 different things that they were looking at when they do their food testing. All right, but we'll save that for another time. So let's keep going. Oh my goodness, the questions keep coming in. All right, here we go. Let's pick this one. All right, how frequently do I need to calibrate I guess that's not flour, but fluoros and thermo temperature, I guess, for my instrument maintenance, except it goes really off. Oh, maybe I shouldn't have picked this one.
I think that's, I think they're asking, how do I need to account for thermal validation and fluorophore validation within a thermocycler?
Beautiful.
I would argue the answer really depends on the type of unit that you have. If it's a bulb-based or laser-based system, those do change. Our systems are LED-based, and they are not really going to change in terms of data output. The ultimate test is coming back to what Adam has been saying, is a standard curve. If you can run a small standard curve with a known target and prove out that it amplifies, that's how we qualify our machines. That's our qualification plate to prove that a CFX machine is in working order. So the answer of what is really off, it really boils down to, can you replicate a known positive standard curve? Can you generate small changes with clear delineation? So there's no hard question, though, we can say, you know, this is when a qPCR machine has become too far off the rails to be used anymore.
Yeah.
Got it.
Absolutely, Brandon. And I would say, sorry, I didn't raise my hand. My bad. I would say, though, that on top of, yes, some machines require yearly or bi-yearly fluorophore calibration, given the type of light emission that they use. The thermal calibration is something that happens much more rarely, and I would say definitely on Bio-Rad machines, which don't require the optical calibration, we very rarely hear of that. The best way to determine it is, of course, to use a standard that works well. And if you start seeing some, drift in that, then maybe you can explore the fact that there is something off in the thermal block. I would say that rarely happens in our machines, but it's always something good to check.
Fantastic. All right, let's keep us moving along here. Next up, ooh, here's a nice one. Is it always best practice to let the PCR machine set the threshold and baseline? The CFX does this, and I like to trust it. So hopefully the answer is yes. Adam.
So yeah, the vast majority of the time it is. There are a few, I have seen a few occasional instances where, an algorithm is an algorithm. It can be a good one, but nothing is ever perfect. So I've seen a few instances where, the software set a threshold, but maybe we had one sample whose fluorescent level jumped up really briefly at an early cycle and passed that threshold before it came back down. And you really saw that it was amplifying, at cycle 20 or something like that. So generally speaking, you can trust where the thresholds are set, but you still want to kind of trust but verify. You want to eyeball each data point and make sure that what you're seeing is in conjunction with the data that are being represented. And so if that's the case, then you can change the number of cycles that are analyzed to establish the baseline. You could use a linear regression model instead, or you could simply move the baseline threshold up a little bit manually so that that isn't the case. But in 99.9% of my experiences, where the threshold has been set has been accurate.
If I may chime in here, I think the subtextual question is, how do I make sure I'm not telling myself a lie? If we set a threshold, we can make that happen. I mean, between the people who are on this call, we know how to get any answer we want out of this machine. Between what you can do with qPCR, baselining, thresholding, if you want to tell yourself a lie, you can with this technology. So we have to be so vigilant in the opposite direction to say that we will not let our personal biases affect the outcome, or it will go wild with that. I mean, it's an incredibly difficult thing to know that you're doing qPCR and having to almost separate yourself from it and say, I need to be willing to accept the outcome, even if it's not what I want. Because the underlying story of the last 20 years is a lot of people have used qPCR to tell themselves comforting things.
Yeah. Yeah, and I think
Brandon,
it's not even. Go ahead, Jan.
No, I love how Brandon put it. I love how you bring it back to, do I want to tell myself a lie? And I feel like there's room for a whole entire discussion about this and about self-truth and how molecular biology can teach us about ourselves as well. So I think we need to schedule another webinar to discuss that part.
I think it was Socrates who said that all I know is that the machine set the threshold.
It was Socrates. It was.
Oh, yes. Back in the day, right? When we all started our PhDs. Yeah, and I think that it could be expanded beyond molecular biology, because I always remember back in the lab days, it's the question you ask, because even the way you ask the question can influence the outcome. So you have to ask that question in just the right way that you can actually get to the scientific result, and to your point, not the result that you want, right? So that you can get your PhD or your publication. So, yeah. All right. Whew, we got a bit sidetracked there, but good sidetrack, I say. Good sidetrack. All right. Ooh, this is a good one for Jan. What are the new instruments coming in qPCR? And I don't know if that means like what's the new instruments, or are there like new versions of qPCR? I mean, we have digital PCR. This is a nice big question.
All right, Derek, I feel like this question comes from you. I don't know. I mean, are you spying on us? First of all, you don't need a new instrument because the CFX is perfection. So that's one. But yes, there will be, at some point, new developments in qPCR instrumentation. And all I can say is just stay tuned. We know that the CFX is a very loved instrument, and we would never dare to come out with a new instrument that did not meet those standards. So yeah, just stay tuned. That's going to be my answer. Why did you give me this question, Laura? This is a very tricky question to answer. I feel like my boss is probably listening.
Yeah, maybe it was your boss who wrote it. No, no, I think it's a good question because, you know, back in the day when I was in the lab, it was always, you know, what's the next best thing? What's the next best thing, right? Do I have the best instrument? Am I missing out on something? But like you say, right, the CFX is a good instrument, but people use it all over the world. It's reliable. You know, if it ain't broke, don't fix it, right? So, yeah.
I mean, there is always room for improvement. I cannot say that, but for sure, we know that at least in instruments like the CFX afford enough flexibility to their users that they have been able to keep up with most new developments in RT-qPCR, and that's really what we've tried to keep up with. We of course keep improving our software, our analysis software, Maestro. And yeah, the instrumentation will also see updates soon. That's all I can say.
All right, moving on. So what is the purpose? Oh, this is a back to basics one. What is the purpose of the calibrator sample when running qPCR? I feel like this should have an A, B, or C answer, right? This is a multiple choice question. Not really, but go ahead. Go ahead.
Yeah, so I guess it kind of depends on what's meant by calibrator sample, right? So if you are running multiple plates and you're looking at different, if you're looking at the same gene on multiple different plates that have to be run at the same time, and you're wanting to compare the expression levels or the quantities of, you know, different samples for the same gene on those different plates, then you need an inter-run calibrator, right? You need at least one sample, the same sample, same assay on both plates to account for any inter-run variability. So in that case, the calibrator sample is just there to account for any differences that may have occurred between this plate and that plate. You know, otherwise, you know, in terms of calibration, it just depends on what's meant by calibration, right? Some instruments require routine calibration because of the way they read the plate, right? You may have an older instrument or some model instrument that has one bulb in a fixed position that's exciting the fluorescence for every well on that 96 or 384 well plate. Each well is getting light at a different angle, a different amount of light, and that needs to be accounted for. And so that's what's done by calibrating the instrument, and that's what purpose that sample and that plate is serving.
Great. Hopefully that addressed the question for our audience member. We didn't give them a name, so let's just call them Bob.
Oh, sorry, Eric.
Eric, there you go. That's a good one. Yeah, well, maybe we could have themed names like different TV shows that we like. Eric makes me think of South Park. Okay. So here is another one. Hopefully you can see this. Why is qPCR less widely used when testing targets in cell-free DNA? For example, rare somatic mutation detection.
I can take that one. And I might, you know, elaborate on the question, Gerald, because I feel that the entire question is why is qPCR less used than potentially digital PCR when evaluating somatic mutation detection in fraction. Sorry, the fraction of somatic mutation detection. So the purpose of said studies is really to determine not just whether certain mutations are present, but also at what level they are present. Therefore, you're analyzing the percentage of a certain mutation over the entire amount of wild-type DNA in circulating cell-free DNA. This is particularly relevant in several types of cancers, and this is definitely what has been leading the field of liquid biopsy. And among the methods that can be used, qPCR, digital PCR, and also now NGS are definitely the three methods that are being published on. Now, I would say the advantage that digital PCR presents over other methods here is that because every reaction is partitioned, you minimize the risks of competition and basically overpowering of the wild type DNA over the fraction of mutated DNA. So let me explain this. So you have your qPCR reaction with one set of primers, two sets of probes, one probe for the mutated allele, one probe for the wild type allele. If you run the qPCR reaction, it is typically acknowledged that you can detect a fractional abundance down to about 1% of that mix. Now, clinical researchers would like to get as low as possible in order to detect, of course, cancers as early as possible. And that's when methods like digital PCR really, droplet digital PCR really provide an advantage. They, by partitioning all the reactions, minimize the competition between that wild type allele and that mutated allele. And you now get the possibility to go even lower at 0.1%, or even in some cases to 0.01% allelic fraction. So that is why I would say qPCR is less favored in those type of applications. Not to say that it cannot be used, and there are actually some fields like leukemia research where it's still a very popular technology. But for the most part, I would say that droplet digital PCR is a better suited technology. Guys, anything else to bounce on that?
Well, I think the word that they added there, rare, is what makes it where you can't really rely on qPCR. If it were simply asking heterozygote versus homozygote, that's something you can absolutely do with a qPCR test because it's either, say, one probe versus two probe versus both. It's very easy to call 50-50 or 100 either direction. But if you're going to ask yourself, can I identify, especially within, say, a human cell-free DNA, an incredibly rare target with a lot of accuracy, you've got to, as you said, partition it out to multiple reactions and rely on the mathematics of that.
Mm-hmm.
Fantastic. All right. So I'm going to have the pleasure of introducing this question from Katrina, let's say. This one, here we go. So this person, Katrina, is using 384 well plates in Quant Studio 5, and the other is in an Applied Biosystems machine. So at the same time using SYBR Green. Can they use the data from the two machines and their CTs into one plot to comprehend the data? Please help. Ooh.
I'll take that.
We love nothing more than to help troubleshoot our competition's problems.
There you go.
I gotta say that.
We're very benevolent.
I would argue this is equally applicable across any two different machines. Even the same manufacturer, the same model, if I were comparing against different machines, as we discussed a little earlier and Adam alluded to with the idea of standard curves, you would need to have some form of either calibrator in the form of a single sample or just simply comparing to a standard curve on both plates. And so if you take a sample that's raw CT or CQ value on a plate, compare that to a known standard curve, and then extrapolate that out to a concentration, that can now be compared across any given samples. But the raw CT or CQ values as a whole, those do not have the ability to directly just be compared against plate to plate, particularly from machine to machine, where detection differences are going to very much be real.
Right.
Yeah. Yeah. I agree. I mean, you have to have some common denominator between machines, between plates. Otherwise, it's really impossible to have a comparison across the plates. Sorry, Katrina.
Brilliant. All right. So this one is from Ashley. I've heard that hydrolysis probes are more specific. Should I be using those instead of SYBR Green? Adam.
Go ahead, Adam.
Oh, yeah. So that is one perception that's out there, is that because SYBR Green is a double-stranded DNA binding dye, that using hydrolysis probes are going to give you a more specific answer it's important to keep in mind that the most important thing in determining the specificity of an assay is the primer design. Right? I can get an assay that's perfectly specific with SYBR Green if I've designed the primers appropriately. And regardless of what detection chemistry you're using, it has to be validated for specificity anyway. So it's true that having a probe, a third oligo, is going to give you another layer of binding that can enhance specificity. But if your assay design is done properly and you validate it, you can use either chemistry and be perfectly comfortable with your results.
Yeah. And I would add to that, yeah, just I would say I know that probes definitely can add that extra element of specificity, but they also are sometimes difficult to design against. And in particular, when people are targeting small areas, you're trying to, you're trying to get a 60 or 70 basis amplicon and try to fit in a 25 base probe in it can make it difficult. And if you have to compromise efficiency for that added level of specificity, I would say that's not necessary. I mean, SYBR Green with the right controls, with the right validation, is a perfectly specific technology. And you should not limit your dreams, Ashley, because people told you that hydrolysis probe are the go-to.
Good PCR starts and ends with the primers.
Right.
Yeah, I wonder, did we come up with SYBR Green because we're Bio-Rad?
No, that existed before that.
Okay, all right.
Yeah.
Just checking.
And we did not pull Eva Green because of the actress neither.
Prior art, though.
Yeah, yeah. All right, so here's another one. This one is from Chelsea, and we're kind of start to wrap things up on this side of things. So with the increased demand of qPCR supplies, what can I do to increase my throughput and use fewer reagents? Are there any downsides or caveats to doing this? Yeah.
I can take that one. Yeah, so Chelsea, I would say this is definitely something that is in everybody's mind. With COVID-19 pandemic, there has been a lot of extra demand from all suppliers for qPCR reagents. You have several tools at your hands. Of course, using 384 well formats instead of 96 can help you get more out of your plates. If you are finding limits in the plastics, etc. Regarding the reagents, yes, you can do reactions with less volume. If you are used to running a 20 microliter reaction in a 96 well plate, you can translate that to a 10 microliter reaction in a 384 well plate. What I would say is if you are performing pathogen detection, in particular COVID-19 detection, you still need to validate your LOD with set volumes. You are now putting in less volume than initially, and therefore you should see if this type of format still gives you the LOD that you desire. So not impossible, but definitely has to be done with some afterthought.
Yeah, keep doing the good science, right? All right, here's one from, I'm going to say this one's from Mike. How do I ensure that my qPCR follows literature and industry scientific rigor? And the Mike is a hint, right?
So yes, the universal guidelines for this thankfully already exist. So within the scientific literature in 2009, there was a set of guidelines, the MIQE guidelines, M-I-Q-E. And those were created in response to a bit of a controversy that we don't have time to delve into, but someone claimed that vaccines caused autism using bad qPCR data with no controls. And what we learned from that is that people will continue to cite bad data long after it has been debunked. And so to prevent that from being the case and to see qPCR used more for good than ill, the people who were the thought leaders within this field created the MICE guidelines, and it says, here's the universal template for how we do good science that is replicable, that is transparent, and can be conveyed to anyone else within publication, and then those people can go and reproduce the same outcomes. So we didn't want to have this constant chase of, oh, I published this, someone can't reproduce it, now I retract it. If we're not meeting this in the first place, it should serve as a greater barrier to asking, well, what controls can we do? All in all, it's really nothing more than a checklist of everything you need to do qPCR correctly, and how we, again, keep ourselves accountable, that we do not tell ourselves convenient lies. And I'll just cite a very recent example of this, of a paper that did not cite did not meet the MICE guidelines and was published as a preprint article regarding COVID-19 and purported to say that masks did not stop the infection from spreading. Well, then it was retracted shortly thereafter with a note that said, oops, we didn't know how to do these calculations. We didn't follow the qPCR guidelines. And, you know, proving the negative on something you're not doing to the fullest extent could be an exceedingly forward statement to make. And I'm not here to scare everyone straight into doing qPCR right, but again, what we can do is to hold ourselves deeply accountable and run those controls and assure ourselves that what we're doing cannot be anything but the answer that we think it is.
Yeah, agreed. I think right now, now it's funny, like people are more interested in science than they've ever been. So we're kind of like the cool kids now, which is fun, right? We've never really been the cool kids, but with all of that comes big responsibility. So I think great points you make there about making sure we're putting out good data, reliable data, so that we can, keep science as held up in as a high standard. Anyone else have anything to add on that last point there? Jan?
Yeah, I do. Thank you. And thank you also, Laura, for mentioning that. All my friends from my Dungeons and Dragons club agree that it's finally time that we get recognition. But the main thing here is to keep in mind, and for qPCR, as with any technology, manufacturers have really been striving to provide simplicity, ease of use, and straightforward protocols to use. But that does not mean that the technique itself is simple. And every technology, even just basic spectrometry, you need to understand what is behind it in order to draw the right conclusions. And the MICE guidelines are basically the example for qPCR. But every technology really has or should have those set of guidelines in order to draw the right interpretation from it. So,
fantastic.
Yeah. That's exactly right. From any technology, you can generate a data point, right? You can draw a square around a blotch on your Western blot and, say it's a band, but, it's really a matter of, is it living up to a standard? And that's what MICE is to give us a standard by which to follow and transparency by which for science to, make sure that we're practicing it in the best way possible.
All right. Well, I think that's a great place to end us for today. So thank you very much to my great panelists this morning. It was an absolute pleasure to discuss these brilliant questions. And thank you to the audience as well for sending in those questions. And before I go, I would just like to do a super quick plug for some of the other coffee chats that we have. Some of them are available on demand, depending on when you're listening to this one, or our upcoming events that you can join live. All types of different discussions, from digital PCR and cell and gene therapies to mixed mode resins for protein purification, cell sorting tips for success. So hopefully everybody really enjoyed it as much as we did. Thank you again, Jan and Brandon and Adam.
Of course. It was a pleasure.
Thank you, Laura.
And with that, I'm going to sign us off. Take care, everybody, and stay safe. Bye-bye for now.